we'll move to the next talk. And this is going to be Dr. Hosseini from Iran, and he's going to talk about the antiproliferative effects, cell cycle alterations, and apoptosis induction of DDW on Z47D and HT-29 human carcinoma cell lines. Dr. Hosseini?
Yes, thank you. All right. Actually, I must confess that I have learned a lot today because I have been exposed to too much diverse information on DDW. As I have mentioned last night, we have been in this business for the last couple of years, and from when we have had access to this DDW. And we have invited medical universities and to work on this DDW. And we have ordered some sort of research activity to the Tehran Medical University, to Dr. Ibrahim Azizi's lab. Actually, I am presenting his slides. He couldn't be with us today. The work that we present here is the antiproliferative effects, cell cycle alterations, and apoptosis induction of DDW on T47D breast cancer and HT-29 colorectal cancer cell lines. I don't need to mention really that how treatment of cancer is important, and that the cell cycle alteration and or decreased apoptosis induction following chemotherapy are primarily due to molecular changes of cancer cells. Studying new compound alone or in combination with potent chemotherapeutic drugs such as Doxorubicin can open new strategic approach in cancer therapy. Previous reports indicated that the potential anti-cancer effects of deuterium-depleted water, mainly from Dr. Sorry, I forgot. I forgot your name. Shumlai. Is that right? Yeah, okay. Okay, so we have been motivated to study the effectiveness of this DDW water in cancer therapy. And we have done this because we have, as I said, we have had access to two types of DDW. We called them A and B. And those days we didn't know which one is what in terms of the content of deuterium. So we have done these cytotoxicity effects, cell cycle alteration, and apoptosis induction studies on those cell lines that I have mentioned in the presence or absence of doxorubicin. In terms of methods, all of those solutions, buffers are just prepared based on what have been explained in literature. For the cytotoxicity experiments, we have used 10,000 cells or for the case of the colorectal carcinoma cells, 5,000, cultivated in 96 well plates. The culture media replaced after 48 hours with DDW A or B in the presence or absence of 250 nanomolar doxorubicin. Cells were then counted at 540 versus 690 nanometers using a sunrise plate reader after treatment with MTT in days 2, 4, and 6 for T47 or at 10 days for the HT-29 cells. And all of the experiments were conducted at least 3 times, and the results are demonstrated as means plus and minus standard deviation. For the cell cycle alteration, we have used 40,000 cells from each, cultivated in 12 well plates, and the culture media replaced after 48 hours with DDW A or B in the presence or absence of 40 nanomolar doxorubicin. Cells then treated with trypsin EDTA washed with PBS buffer, centrifuged and homogenized a DAPI cocktail and incubated for 30 minutes in dark. Cells then transferred to flow cytometry tubes and the fluorescent intensity read and recorded at FL4. Data analyzed and the percentage of cells at G0, G1, S or G2M phases defined using FluMax program. And for the apoptosis experiments, we did exactly the same, but we treated the cells a little bit different. And then when we transferred the cell for flow cytometry, and the fluorescent intensity read at FL1 versus FL2 and recorded for each quadrant. Here are the results. Actually, this is supposed to show and demonstrate. What do they do? Okay. Oh, yeah. Cytotoxicity effect of DDW alone on breast cancer cell lines using MMT methods. This is the culture media at 2 days, 4 days, and 6 days. And these, then the cells are treated with 250 nanomolar doxorubicin with normal water. And these are the media which have been prepared with DDW A and B. As you could see, there is not much difference really between the normal water and DDW water in terms of the cell counts and the cell toxicity when we are using the DDW alone. But when we studied the cytotoxicity of DDW in combination with doxorubicin on breast cancer cell lines, it seems that there is slight sort of increase in the effect or the toxicity of the doxorubicin when we use the doxorubicin in DDW-A or DDW-2B media. And this is the results for the other cell line, colorectal cancer cell line, doing the same thing. Again, it's very similar pattern to what we have seen for the other one. The culture media alone, whether it has been ready with the normal water or A or B DDW B water, doesn't make that much difference. However, there might be a tendency of increased cytotoxicity if we would have put, if we could expose the cell lines for a longer period of time. This is the study, the cytotoxicity effect of DDW in combination with doxorubicin on HT-29 colorectal cancer cells. Very similar pattern as we have seen for the breast cancer cell line. Here are the apoptosis induction experiments, the result of that, alone or in combination with doxorubicin on T47 cell lines using AnnexiV-FITC or PI method. As you could see, these three bunches of histograms are culture media made with normal water, with DDW A and or B. They don't. It doesn't seem that there are that much difference in terms of induction of apoptosis. But when we use and make our media, our culture media, in the presence of 14 nanomolar doxorubicin with DDWA and DDWB, this, which represents the apoptosis, increases significantly in terms of statistics from that or that. And again, here, this, which represents the necrotic effect of the DDW and the doxorubicin in the presence of those media, it seems that there is a tendency for increased effect of the DDW for these cell lines. This is exactly the same pattern and same experiment with the other cell line, H. Sorry. This is the study, the cell cycle alteration by DDW alone or in combination with doxorubicin on T47 cells using DAPI method. Let me see what I have to say on this slide. Okay. As you could see here, these long histograms, for T47 cell lines, the accumulation of the cells are mainly in G2M phases, based here. Okay. And for the other cell line, for the colorectal cell line, we see that we see a different pattern here, and I think I made a mistake. I think that's right. Yes. Yeah, actually I missed a slide here in my new version. It looks like that. To summarize the results, we would say that both types of DDWs, A and B, showed no apparent effects on proliferation of T47 and HT-29 cell line. Antiproliferative effect of doxorubicin was slightly increased when prepared in DDWs in comparison to doxorubicin alone. The S phase in cell cycle pattern of T47 cells was significantly increased only in doxorubicin prepared in type A of DDW. In HT-29 cells, the G2M phase of cell cycle was significantly increased in both DOX A and DOX prepared with DDWB in comparison to doxorubicin alone. The T47 cells exposed to DOX A and DOX B in comparison to doxorubicin alone showed increased apoptosis and necrosis, respectively. In the HT-29 cell lines, the G2M phase of cell cycle was significantly increased in both doxorubicin A and doxorubicin B in comparison to doxorubicin alone. To conclude, our data supports the previously reported findings on significant potentials of deuterium-depleted water on cancer cells to enhance the effectiveness of chemotherapy. And I thank you very much for your attention, and if you have further questions or you need further explanation, you could contact Dr. Azizi at his email address. Thank you very much.
So the talk is open for questions. I'd just like to ask you one question. It's nice to keep the investigators blind. So the talk is open for questions. I'd just like to ask you one question. It's nice to keep the investigators blind, but the audience is still blind. Do you know what the ppm concentration in A or B was now?
Yes, actually, yes, I've got them here. The A was 50 ppm, and the other one was 105 ppm.
So A was 50, and
A was 50 ppm,
B was
and
105.
the B was 105 ppm.
It seems it was a triple-blinded study.
It was a
now
blind.
audience-
Actually, yeah, it was. It was.
So, any questions, though? Go ahead.
Do you have any, let's say, idea on the mechanism of doxorubicin which might be enhanced by the presence of...
Actually, I personally don't have any idea.
Oh.
You better ask Dr. Azizi.
Right. Okay.
I don't
Thank
know.
you.
Really.
Actually, doxorubicin is, I believe they used it as an antibiotics first, and then it had the anti-proliferative effects, and it by default, it seems that it, as far as we know, because we did profile doxorubicin with some other anti-cancer agents, it increases the oxidative branch of the pentose cycle. So what I would assume, there's a relative compensation on cells adapt to transcurase inhibition by upregulating the oxidative branch, and if it's consistent with DDW somehow decreasing or inhibiting this proton exchange in the glucose 6-phosphate dehydrogenase reaction, that can be one mechanism. But this is just a contemplation, and Dr. Sisi might be able to answer it best, but this would be my take on any mechanism that is involved.
I would like to some short note because I am thinking, so finally you repeated that checking the cell growth after 24 hours, you couldn't confirm that the deuterium-depleted water itself can inhibit the cell growth. And one question is, which could be common with your experiment, the time frame that it's 2 days or even longer. But the other question came to my mind that when you put the cells into the media and put it in the chamber, keeping on 37 Celsius with a 100% humidity, do you try to avoid the exchange between the media and between the humidity? Because if you don't,
we didn't.
Okay,
No.
if you don't do that, finally, gradually the deuterium concentration will increase in the media, and you are not testing a media with low deuterium level. And I don't know whether you try to avoid it. Not close, because there is an exchange, because the pH is regulated and the carbon dioxide is going and the water. So that could be, I think, a disturbing factor. Okay, just came to my mind because I know I was sure I also tried to avoid any exchange between the air, so that can be a disturbing. Okay, thank you.
Okay, thank you.
All right, so thank you, Dr. Hosseini. So